Hi guys, I am trying my best to get to Alaska as soon as possible, but before I get there, I have created this video and a concept map to guide us in determining what this thing may be. The first thing we are going to test for is if it contains the 4 biomolecules. To test for proteins, construct a burette test. In this chemical test, take the sample and add an equal volume of a strong base. I would recommend the base to be sodium or potassium hydroxide. Then add a few drops of aqueous copper 2 sulfate. If the solution changes from blue to pinkish purple, then there are proteins present, and if the solution turns to pink, then there are short chains of polypeptides present. The shifts in color happen because the copper atom of the burette solution reacts with the peptide bonds. To test for lipids, construct the grease spot test or the Sudan strain test. I would recommend the grease spot brown paper bag test because it is simpler to construct. Put a few drops of the unknown sample on a brown paper bag and watch what happens through time. If there are translucent marks, grease spots, left on the unglazed brown paper bag, lipids are present. If there are no marks, there are no lipids. To test for carbohydrates, I would suggest constructing Benedict's test. Put 1 milliliter of the mysterious solution in a test tube and add 2 milliliters of Benedict's reagent, a blue-colored liquid with copper 2 ions. Then, heat the solution in a boiling water bath for 3 minutes. The formation of a reddish-orange precipitate within 3 minutes indicates there are carbohydrates. This reaction is caused by the reducing property of simple carbohydrates. The copper 2 ions are reduced to copper 1 ions. The test is negative and the solution does not contain carbohydrates if after 3 minutes the solution is still a blue turquoise color. To test for nucleic acids, construct a diphenylamine test. In this test, combine the substance with diphenylamine. In the presence of DNA, diphenylamine is oxidized and the solution will turn a clear blue. The more DNA present, the darker the color. RNA will turn the solution green. So now that these tests have been conducted, does it contain the 4 biomolecules? If it contains the 4 biomolecules, then it is either a prokaryote or eukaryote cell. If it does not contain all 4 biomolecules, skip over the next few minutes of this video. Prokaryote is defined as a microscopic single-celled organism that has no nucleus nor other specialized organelles. Eukaryote is defined as an organism whose cell contains a nucleus surrounded by a membrane whose DNA is bound together by proteins. To tell the difference, look into a microscope, and if you see a nucleus, more than 1 chromosomes, endoplasmic reticulum, mitochondria, and Golgi, then it is a eukaryotic cell. If there is no nucleus, no endoplasmic reticulum, no mitochondria, and no Golgi, then it is a prokaryotic cell. Prokaryotic cells are smaller with a typical size of 1 to 10 micrometers. Eukaryotic cells are bigger with a typical size of 10 to 100 micrometers. There are 7 main organelles found in the eukaryotic cells that have specific functions and ways to identify. The nucleus is a highly specialized organelle that serves as the information processing and administrative center of the cell. It has 2 major functions. It stores the cell's DNA and it coordinates the cell's activity. The mitochondria are rod-shaped organelles that can be considered the power generators of the cell. They convert oxygen and nutrients into adenosine triphosphate, also known as ATP. The endoplasmic reticulum, shortened to ER, is a network of flattened sacs and branching tubules that extend throughout the cytoplasm. It functions as a manufacturing and packaging system. Connected to the ER, the Golgi apparatus is a series of 5 to 8 flattened disc-shaped membrane-covered sacs called cisternae. As many as 60 cisternae may combine to make up the Golgi apparatus, giving it a relatively large size. The Golgi apparatus stores, modifies, and packages proteins, as well as making sure each protein goes to the correct destination. The cell membrane is a biological membrane that separates the interior of all cells from the exterior environment. Its function is to protect the cell from its surroundings and control what goes in and out of the cell. The ribosome is a complex molar machine that serves as the site of biological protein synthesis, converting genetic code to protein molecules in both prokaryotic and eukaryotic cells. The difference is that the ribosome in eukaryotic cells are larger than those in prokaryotic cells, and in eukaryotic cells, ribosomes particles attach to the membrane of the ER. The chloroplast, only found in plant cells, is a cell organelle that contains chlorophyll and produces energy through photosynthesis. Photosynthesis is the converting of light energy to energy stored in the form of sugar and other organic molecules that the plant or alga uses as food. Now, if it does not contain all four biomolecules, is it a virus? It can be a virus if it does not contain carbohydrates and does contain nucleic acids and lipids, and possibly proteins. A virus is a microscopic organelle and can only replicate within living cells of a host organism. They depend on cells for metabolism and reproduction, therefore not considered true life. Viruses can only infect their host cell. The typical size of viruses are 20 to 400 nanometers. How would you identify viruses? To test a virus, bring a host cell. Hosts are anything and everything, but I would recommend a plant cell. Look through the microscope, and if it does not contain cytoplasm or organelles, no chromosome, just RNA and DNA, and covered in protein coat, it is a virus. Now that we have determined if this thing is a prokaryote, eukaryote, virus, or nothing, we still have a few questions to answer in regards to prokaryotes, eukaryotes, and viruses to determine whether or not this thing is life. Does it interact with the environment? Place heavy water, H3O, next to it and observe through a microscope. Does it go toward it or away from it? Does it absorb it? If the water does move towards it, it does interact with its environment and is considered life. If the water moves away from it, then it does not interact with its environment and is not considered life. What I have explained with the water can also be done with a lamp, food, and heat. For viruses, offer different hosts such as bacteria, plant cells, and mouse cells. See if it interacts with the host. Does it respond to the host? Does it realize the host is dying and reproduce more? Does it reproduce? Prokaryotic cells reproduce by a process that is called binary fission. This process starts with the replication of the chromosome. The new chromosome attaches itself to the plasma membrane, and the two chromosomes migrate to opposite ends of the cell. The plasma membrane in the middle of the cell grows inward until it closes to separate the cell into two compartments, each with a full compartment of genetic material. The cell then fissions at the center, forming two new daughter cells. A eukaryotic cell duplicates its chromosomes within the interphase. During the cell division phase, the nucleus divides in a process called mitosis, and then the divided nuclei are established in separate cells in a process called cytokinesis. Viruses can only reproduce in living cells. Repeatedly test this by checking for an increase in numbers. If there is, then the cell is able to reproduce and is considered life. Is it complex? Any life form is complex. Single-cell organisms are complex, however, viruses are most likely not. Does it grow? Most prokaryotic cells grow slowly because nutrients are limited in natural environments, and some grow rapidly when nutrients become available. Eukaryotic cells' growth is usually a continuous process through the 4 phases of the cell cycle. Viruses do not grow on their own, only when they have a host. To measure growth, look through a microscope and examine the current size. Then feed it and continue to study the size. If it gets bigger, it is growing. Does it maintain homeostasis? In order to maintain homeostasis, negative feedback loops act to oppose the cue that triggers them. It is maintaining homeostasis when temperature is kept the same as the room temperature changes, keeping the concentration ions balanced depending on the pH and pOH of the substance. To test if it is maintaining homeostasis, take the initial temperature of the foreign substance. Then change its environment's temperature, and again, take the temperature of the foreign substance. Did it stay the same? You can also put it near a heat source to observe whether it moves away or towards the heat to maintain its body's temperature. As I said in the beginning of this video, I'm trying my best to get to you guys in Alaska. Thanks so much for watching this video and performing these tasks to help determine if it is life or not.